Hi, I'm Priti Balchandani. It's a pleasure to be here today and an honor to be invited to this. It's really great that scientists and artists and scientific artists are converging in one spot because I do think science and art are heavily intertwined and enhance each other. So I'm really happy that this is happening. My talk is about visualizing the living human brain, which we do here at our institute. And I am the Associate Director of the BioMedical Engineering and Imaging Institute, housed in the building that we are in. We have our offices on the first floor, and we have our scanners in the basement of this building. I also direct neuroimaging research here at Sinai, so that's why I'm going to talk about the brain a lot. Here are some relationships that I need to declare. I'm an inventor on some patents and investor in a biologically guided radiation therapy company. So, as I was describing earlier, we have a suite of research MRI scanners here, and we use this to visualize the human body, this set of scanners, in unprecedented detail and through different filters and contrasts, and we can look at several aspects of physiology and anatomy. And today I'm going to focus on the images that can be provided by a very high field or high magnetic field MRI scanner, which operates at 7 Tesla magnetic field. So clinical field strengths are 3 Tesla and 1.5 Tesla, and here we have a 7 Tesla whole body MRI scanner, again in the basement of this building. And the building on the left is artist rendition of this building that we're in. So I thought I could start with talking about the multimodal MRI at 17 and what we can actually see. And it's remarkable the kinds of things we can see in vivo, non-invasively, in the human brain, just using this one device, the MRI scanner. And then I would like to provide an example of one clinical application, or several clinical applications in the skull base, so skull base pathology. Here I will comment on the value of medical illustrations. Many of the artists that we work with are here and organizing this conference, and I wanted to show you how intricate anatomy and effects of disease and circuits that are affected in the brain can be shown using MRI, but then explained using scientific visualization or scientific illustrations. And lastly, I'll close with a gallery of images that we have created, or the team here has created, where art has emerged from medical images. And these were actually displayed in a art gallery here and in the neighborhood. And we also created a calendar for using these images. So I thought that that would be a nice way to close. So let me begin with multimodal MRI of the brain. This is the main objective with MRI, or the most common type of image we think about, and that is a image of anatomy, a very high resolution image of the structure. And at 7 Tesla, like I talked about earlier, we can see this anatomy in really exquisite detail. On the left, you can see the gray-white juncture is really nicely visualized. And on the right, I'm zooming into a structure in the brain called the hippocampus, which is a small region in the brain that is involved in many different things, including memory formation, and is affected by a wide range of diseases and can cause symptoms for a wide range of diseases, including epilepsy and stress-related disorders. It's a seahorse-shaped structure, and it is composed of several subfields or subregions, and each one of these are separate sensitive markers of disease and are functionally different. And at this resolution, we can actually differentiate these subfields and delineate them and evaluate the or quantify the volume of these subfields, which becomes a nice quantitative marker for disease processes. Beyond anatomy, we can actually look at vasculature. So arteries that feed the brain can be visualized. And how do we do this? Well, the way MRI works is that we are getting signal from the water in our brain or in the body. I'm too focused on the brain, so you'll notice that. So the water is what creates the signal, and that's how we create these images. And blood in the arteries is moving faster than the stationary background tissue. And we're able to differentiate the moving spins or moving water molecules in the blood versus the stationary water molecules. And this again becomes enhanced when you increase the magnetic field strength, because the moving spins retain their magnetization for longer. So we can see tinier branches of the main arteries that feed the brain, such as the carotids. We can also see veins, and we use a different mechanism to see veins. Here you can see tiny veins in the cortex. The reason why these become easily visualized is because veins contain deoxygenated blood, which is paramagnetic in nature, so it has an iron core that becomes exposed when the hemoglobin becomes deoxygenated. And so this creates a difference in the magnetic signature and a dephasing of the signal, or the loss of the signal in the veins compared to the surrounding tissue, and we can leverage that to create these venograms. Then we can use image processing and image analysis algorithms to segment these structures. Here we're segmenting the veins, and it is done automatically. And after you segment, you can actually quantify the numbers and the volumes of these structures, in this case the veins. And then we can look at them in specific regions, like shown here on the right, in a healthy patient versus an epilepsy patient, when looking at the veins in the hippocampus, like I talked about earlier, and the adjacent amygdala. An additional type of contrast that we can perform is called diffusion MRI. And in this case, we're able to look at the diffusivity of water molecules. The diffusivity of water along fiber tracts. What are fiber tracts? Are bundles of axons that connect up the brain. So basically the structural circuits within the brain. So the diffusivity of water along those axons is different than the diffusivity of water across those axons, or the water is diffusing more rapidly along axons versus across axons. And we use this simple mechanism to actually map out all the fibers or bundles of axons that connect up the human brain. On the left are, so these images actually show graphical representations of this diffusion tensor, or this, the diffusion tensor describes the diffusivity along the different spatial dimensions. And we are able to characterize that diffusion tensor in very high resolution. 7 Tesla provides even more granular resolution for this type of contrast. The colors represent the direction in which the fibers are. And so we can take this information and create tractograms of the entire brain. Here I'm showing it in the 3 different orientations. And again, this allows us to see all the main neuronal fiber tracks that allow the different brain regions to communicate with each other. We can isolate these fiber tracks to specific structures that we're interested in. Here we've looked at the tracks emerging from a part of the brain called the amygdala, which is involved very much in emotional response and emotions. So we study this in several diseases, but specifically psychiatric illness. We can also look at those diffusion tracks coming from subregions of the hippocampus, like I talked about earlier. So because of the resolution imparted by this high magnetic field scanner, we can actually look at the tracks emerging from the subfields of the hippocampus, and this is the first time this has been done. After we have the entire whole brain tractogram, it's possible to take that as well as other information on the functional activation of the brain, segment the brain using automated methods, segment the anatomy of the brain using automated methods, as shown in the center here, and then create connectivity matrices that describe quantitatively how connected certain regions of the brain are. And we can also use graph theory to try and understand how the network properties of the brain are changing when it comes to disease versus health. This is a connectome that has been created from that diffusion data, specifically for this subject who had scans that happened to be performed prior to a head injury and after a traumatic brain injury. So we actually took a look at how this connectome changed prior to the injury versus after the injury. We can also actually look at molecules other than water that contain hydrogen, and this is called MR spectroscopic imaging. So we can look at a spectrum of these metabolites. And here we are showing spectra emerging from, again, the area that I was talking about earlier, the hippocampus. And these metabolites give you an idea of neuronal health, neuronal density, and neuronal function. So that's some of what we can do with one device, an MRI scanner. And there is no invasive procedure required here. MRI is non-invasive. So I'll talk about how we apply this in one application, which is skull-based pathology. And I'll start with skull-based tumors. These are tumors that arise in the area of the skull, the base of the skull, in which there's the bony ridge in front of the in the front near the nose, and then there's bones that comprise of the base of the head. And there are many lesions that actually arise in this region, ranging from pituitary adenomas, meningiomas, and craniopharyngiomas. These are often not malignant, but can be, and cause compressive symptoms such as vision loss. And vision loss is one of the main neurological functions that are impaired in these tumors. There's also hearing loss, facial pain, and numbness. So they are accessible through different means when performing neurosurgery, and we can alleviate these symptoms through resection of these tumors, but they are close to some very intricate and complex nerves and arteries. So the cranial nerves are often compressed by these tumors or encased within them. the cranial nerves that are involved in vision, and specifically the optic chiasm, is often interacting with these tumors. And there are also vessels that are branches of the carotid arteries feeding the brain that are compressed by these tumors, are adjacent to these tumors. So because of this complex region and all of these intricate structures that are very delicate surrounding these tumors, resection is actually quite complex, because damage to these structures will cause cataclysmic consequences. So there's the possibility of very severe complications if damage occurs to those structures. This is why MRI plays a big role in performing these surgeries. And in the past, the surgeries have been very invasive and required the removal of the skull. But recently, or in the modern approach to this surgery, endoscopic methods are used, and an endoscope is inserted through the nasal passages and illuminates that cavity in the skull base so that these tumors can be accessed entirely through the nose, and there is no scalp removal or skin incision. The trauma is much reduced, and there's decreased morbidity and mortality. But as you can imagine, the view of just an endoscopic camera is not going to be very, very detailed when performing these surgeries. So again, our visualization using MRI plays even more of a role. This is a pituitary adenoma, a very large pituitary adenoma that we imaged using 7 Tesla. And here, the tumor itself has heterogeneous regions, which are more clearly visualized using this magnetic field strength. So regions of firmness and softness, vessels within the tumor, draining veins, feeding arteries, compressed mammillary bodies around the tumor, compressed optic chiasm, which is one of the main components of the optic apparatus. And we looked at it in 2 different types of contrast, T2, T1, and then we're able to perform that time of flight angiography I was talking about earlier, which allows us to visualize arteries. And what can be done is, for example, in this different tumor here, a meningioma, that time of flight image can be overlaid on your underlying structural image, and as shown on the right, the tumor itself is marked with an asterisk, and the time of flight image is overlaid with a heat map to show those vessels very conspicuously. Now, these images actually are used in the operating room in addition to the endoscopic camera image as shown on the left and in the middle. So the endoscopic camera really just shows a pulpy mess, but the surgeons are able to differentiate tissue types from that, although they really need the guidance of a co-registered MRI image. And they register their instruments, the endonasal endoscopic instruments, using infrared markers that are placed around the bony anatomy, and then register to the high-resolution MRI image in order to guide their surgery. We evaluated in 20 patients whether 7 Tesla improved this, and in many, there was statistically significant improvement in the visualization of cranial nerves and arteries when performing this surgery. Now, beyond actually performing improved planning and guidance for surgery, we looked at different circuits and systems in the brain that were damaged because of the presence of these tumors. And because of the complexity of these systems and the interplay between different circuits of the brain and regions of the brain, as well as the intricacy of the anatomy itself, it was very important to work with scientific illustrators in order to convey the findings for these studies. So the first study was to take a look at what kind of damage was occurring to the entire optic apparatus because of these tumors existing in the skull base. And we not only looked at the region surrounding the tumor, which included the optic chiasm, which is in the center here, but we looked at cortical thickness for the primary visual cortex, which is in the region of the brain far from the skull base. And we looked at the optic radiations as well as optic pathways through diffusion tractography, like I talked about earlier. So this work was performed by Jack Rutland, and he has worked a lot with the medical illustrators here at Sinai, showing what type of system he wants to target. Here is a graphical representation of all of the structures involved in vision, including the eyeballs, the optic pathways, the optic chiasm, the optic tracts, and as well as the optic radiations that connect to the primary visual cortex. We did find damage, statistically different integrity of these tracts when compared to healthy controls for patients with pituitary adenomas. Here's another system that was studied and an example of how this was illustrated. This is a very small region called the lateral geniculate nucleus that was studied. It is part of the thalamus, which is one of the main relay stations in the brain for sensory and motor signals. And specifically, we wanted to study with these patients with pituitary adenomas whether or not the size of this structure, the LGN, or the lateral geniculate nucleus, which is within the thalamus, was different, and whether it correlated to recovery of vision after surgery, which allows us to get a sense of prognosis. So as you can see in the zoomed in figure on the MRI, the LGN is actually denoted by the white arrow. And we are able to differentiate and delineate this structure, but as you can see, it's not very easy to really just convey or visualize what we're looking at here. We're able to see it at this resolution at 7 Tesla. And that is one of the reasons we did perform this study, is because for the first time we're able to evaluate the size. And this illustration, which was just very comprehensive, created by Jill Gregory, allowed us to allowed Jack Rutland, the author on this, to depict the reduction in size of this LGN on the tumor, due to the tumor, and exactly what region we were studying, exactly what effect we were looking for, and what the anatomy looked like around this pituitary tumor, and exactly what compression of structures was being evaluated, what structures were being evaluated and how they would be compressed. So this was a beautiful image that basically worked very well to communicate this idea. There was a reduction in the LGN. This has been published. The other study that was performed was very interesting, again by Jack Rutland, and he basically was interested in mapping out the optic, the primary visual cortex, the thickness of the primary visual cortex, which is shown here on the left in an MRI image, and creating a correlation with defects or impairments in the visual field as was evaluated through neuroophthalmology. And there is actually a one-to-one or a mapping between the defects or the areas of impairment in this visual field and the specific areas in the primary visual cortex. And it is shown here in this illustration by Jill Gregory how those areas in the visual field and changes in those areas correlate to the cortical thickness in the primary visual cortex. She's also showing here the compression of the optic chiasm and how that is the reason why some of this damage is occurring to the entire optic apparatus, and how all of these structures connect to each other in the brain, the chiasm, the LGN, the primary visual cortex. And this complicated study, which yielded the result that there is actually a mapping between the cortical thickness and impairments in the visual field. This complicated experiment with interplay of all of these structures and this exact measurement that is being made and compared was only possible to convey in this visual form, and it was key and critical to this entire experiment and communicating this experiment. Here's another study on the surrounding structures around the pituitary adenoma. On the left here in the MRI image, we're showing where we're sampling the signal intensity. In the center, we're looking at the pituitary stalk and how it may be deviated because of the presence of the tumor. On the right, we're delineating the pituitary adenoma as well as normal pituitary gland. Again, all of these changes, these surrounding anatomy around the pituitary tumor are so well explained in the graphical illustration that was provided for us by Jill Gregory. So that's just a nice overview of the tumors and how we studied these not only for improvement of surgical care, but understanding what's happening in the rest of the brain and how the circuits at large are affected by the presence of a lesion in one portion of the brain, and how recovery can be mapped by looking at markers in the rest of the brain. So trigeminal neuralgia is another skull-based condition that we studied, and this is actually very different than skull-based tumors. It is a chronic pain condition where there is shocking facial pain caused by some damage to the trigeminal nerve, which is one of the cranial nerves. And there's very little, there's a lot to be learned when it comes to the etiology of this disease, what is causing the pain, the source of the pain. It is misdiagnosed often. The treatment course is difficult to map out because several different reasons can exist for this condition. But it is highly localized to the trigeminal nerve, which is one of the main nerves associated with facial sensation, and specifically in the area in which it emerges from the brainstem or is entering the brainstem from Meckel's cave. And in this region, there can be several things that can occur. There can be a lesion, like a tumor. There can be injury, causing some damage to that nerve. There can also be multiple sclerosis causing degradation of the nerve sheath. But very often what happens is that there is a vessel in the vicinity of the nerve that's impinging upon it and causing this pain to occur. If we can see that neurovascular compression, which is the vessel impinging on the nerve, then we can actually perform an intervention called microvascular decompression. And these patients who suffer from severe shocking pain, and often this is called the suicide disease because of this, could potentially have recovery of that from this disease and complete improvement of their symptoms. So if we can find that offending vessel, as we have noted here in this 7 Tesla scan of the skull base that shows the nerve and the vessel quite nicely, then it is very helpful. And this is a graphical illustration by Nika Ford, where the area of interest, the area of study, the specific area of study is really nicely delineated, really nicely communicated, and we have correlated it to our MRI images when performing this. So this work is being performed by Judy Alper in my lab, and she is looking at the trigeminal nerve for a series of patients that were studied compared to healthy controls, and exactly what is going on. What, is there neurovascular compression, or is there some other effect? Diffusion tractography can help us understand the integrity of that nerve and see if there's some sort of damage to that nerve that is causing the symptoms, and how this could actually differentiate patients of a certain type of trigeminal neuralgia from another type. And so this is kind of an illustration of all of the different anatomy playing a role, and that very, very important but tiny region where so much could be going on and where the source of pain often lies. And so explaining what's going on there, looking at the different branches of the nerve, seeing the exact geometry of that area is highly relevant in trying to understand this condition and understand the MRI, as well as communicate what's going on. So that's performed here again by Nika Ford. And you know, the other thing we thought would be interesting to do is to merge the diffusion tractography with the graphical representation of the brain, again by Nika Ford. And this by Judy Alper. She's performed the tractography and showed it in 3D on the MRI of the brain. So this, I believe, would be highly relevant for improved diagnosis, and as well as having better understanding of the mechanisms of conditions associated with pain. I just want to show here a combination of the vasculature as well as the tractography of these nerves shown in green. And here we can get a 3D view of how the nerves and vessels are interplaying with each other. And this is quite possible using the 7T multimodal imaging like I was showing earlier. On top of that, we've done the same thing here. In addition to understanding how MRI can help treatment and diagnosis, we want to look at the circuits that are damaged by this condition. Overall, what is the effect that's happening on the entire brain? And here we study the tracks that connect the thalamus, that relay station, with the somatosensory cortex, which is part of the cerebral cortex that is involved in facial sensation and other inputs, other sensory inputs. And we wanted to study these thalamic somatosensory tracts in patients with trigeminal neuralgia that have chronic facial pain due to the damage to the nerve, which is shown here in this graphical representation. And we did indeed find a reduction in the integrity of these tracts on the affected side of patients with trigeminal neuralgia compared to controls. So that's kind of the way that we have taken advantage or leveraged the idea of scientific illustration in order to not only describe what's happening in medical images, but also describe the kind of experiment that we want to perform and what we want to study, the quantitative markers of disease and intricate anatomy that are involved in these specific conditions. So now I just want to end with the fact that we find the images that we obtain so inspiring that the members in our institute have created a gallery of art in the brain, and I'm just going to show you a subset of those. This is anatomical brain images, the T2-weighted images. What's happened is that Rebecca and Gaurav have used an edge detection filter in order to get the edges of the anatomy, and then added a color map and created this grid. This is formed from a T2-weighted image, a spin echo image actually of the spinal cord or spinal canal, And this is by Alan Seifert and Rebecca Feldman. This is formed from one of those venograms that I showed earlier, that created from a susceptibility weighted image. And again, there has been some replication and colorization. This was in our calendar for the month of October because it had a Halloween-y feel to it. And then this is a picture of the connectomes like I was showing you earlier. Remember, the connectomes allow us to see how connected up the circuits are and if there's any damage to those. So we found those quite interesting as well by Yael, Jacob, and Rebecca Feldman. We also sometimes come across non-humans, and this is a whale. We had a sample of a whale's brain, or actually the entire whale brain, and we imaged that, and also were inspired, Rebecca and Gaurav were inspired to create a art out of that. And this is a image of a brain within a brain, created using the susceptibility weighted image that I talked about earlier that creates the veins, and then also a very high resolution anatomical image. And then of course different color maps on that. Edge detection used to create this, maze-like appearance, and this is by Gaurav Varma. These are actually perivascular spaces, and those are tiny fluid-filled spaces around vessels that become visible at the resolutions that we work at, and they could be markers for inflammation. We're actually exploring those, and those tiny perivascular spaces have been automatically detected and colorized to create this this image here by Gaurav. And Stephanie Brown and Rebecca Feldman took the connections coming out of the amygdala that I showed earlier, the tracks coming out of the amygdala, and the ones that connect to the hippocampus, and have created these sort of flywheels out of them. And this is how we kind of just play with the images a little bit, and create what we think is beautiful art out of it. So I just wanted to acknowledge the team. Everything I presented is from them, and my collaborators and funding sources. Thank you very much for having me.